Good morning, or possibly good afternoon, and thank you for the opportunity to speak today at this Writing the Futures of Infectious Disease event. I'm Katie. I'm a postdoctoral fellow in the lab of Pardis Sabeti at the Broad Institute, where I've been applying metagenomic and in particular targeted enrichment approaches for the detection and surveillance of viruses, both here in Massachusetts and in West Africa, and I'm delighted to be able to present some of that work to you today. I'd like to begin in Arua, in Ede State in Nigeria, where in late 2018, our partners at the Arua Specialist Teaching Hospital, shown here by a yellow dot, identified a cluster of cases, mostly children or teenagers, with severe disease of unexplained cause. This hospital is a centre of excellence for Lasse fever and performed tests for this, as well as for other possible causes for which they had tests on site, including malaria and Ebola, but these were all negative. Having exhausted all the tests they had to hand, they quickly sent some of the samples to partners at Redeemers University for metagenomic sequencing and other tests. In just a few days, the team at Redeemers were able to identify yellow fever virus as the likely cause of this particular outbreak. They did this by metagenomic sequencing, roughly speaking, sequencing all of the genetic material in the sample, and the portion here identified in green as those reads that came out as yellow fever virus. Moreover, because they used sequencing-based approaches, they were not only able to identify the likely causal agent, but through assembling its genome, identify other features, such as the fact that these sequences from the cases around Arua were genetically very similar, but they were genetically distinct from all previously sequenced isolates of yellow fever virus in Nigeria, which are shown at the top of this time tree here, suggesting that the recent reemergence of this disease is associated with a different, newer introduction of the virus. This highlights one of the great strengths of metagenomic sequencing, the ability to identify and assemble a complete genome of a pathogen without any prior knowledge of what it is. And this is particularly important when we think about viral fevers, because the clinical presentations of these diseases are often very non-specific, although the causal agents might be very different. But I think this figure also shows one of the limitations of this approach, which is that a very small proportion of all the reads we sequence are from the virus of interest, or from the causal agent. And this is, in a place with a high throughput sequencing capacity where you can use a NovaSeq and reads are relatively cheap, the option to sequence to a higher depth might be the more cost-effective solution. But in places where you're using a lower throughput instrument, in this case this was a MiSeq, that might not be feasible. There are a range of different sequencing solutions available to us when we want to identify a pathogen and assemble its genome sequence. Most of these involve the same broad steps, roughly taking a patient sample, this is often either blood or a respiratory sample, extracting RNA, converting this to cDNA, and turning that into a sequenceable library. If we consider this on a spectrum, metagenomic sequencing is sort of at one end. This is an unbiased approach where you take and sequence all of the DNA or nucleic acid material in a sample. At the opposite end, we have amplicon sequencing. These approaches can be very fast and very sensitive. For example, these have been the dominant technique used to sequence and generate over 1.5 million SARS-CoV-2 genome sequences to date. But as they use PCR primers for amplification of specific sequences of interest, this limits the breadth of taxa that can be detected in any one assay. And in the middle of this, we sort of have targeted enrichment approaches, and we turn to these in order to balance both increasing sensitivity whilst maintaining a certain breadth of detection. These approaches, instead of using primers for specific amplification, use biotinylated probes that hybridize to complementary sequences of interest that can then be captured while non-bound fragments are washed away. As these probes are more tolerant to mismatches than primers, and they can be mixed in greater numbers in a single reaction, this type of method is more amenable to detecting a diverse set of sequences. Indeed, these approaches had already been used by several groups for enrichment of either a single or many viral genomes. However, the probe sets were often ad hoc, thus not readily updated, or proprietary. Now, given the genetic diversity of microbial sequences, and the need to balance capturing diversity with cost constraints and reasonable probe numbers, as well as the fact that more diversity is continually being discovered all of the time, we felt that there was a need for a new probe design approach, and specifically one that had three main features. First, it was flexible to any set of input sequences, that it would be scalable to altering probe numbers or properties, and third, available and easy to run on new sequences as these were generated. The approach that we developed, named CATCH, which stands for Compact Aggregation of Targets for Comprehensive Hybridization, started out as an effort to design the best set of probes to optimally cover all viral genetic diversity within a reasonable budget, and became a freely available tool to guide intelligent probe design. The way that Catch works is given a desired number of probes and an unaligned set of genomes that you wish those probes to cover, Catch will identify the optimal set of probes to fully cover all of that diversity within the input genomes, and in a number of probes equal to or less than your target. To do this, it first determines the fewest probes required to cover all genomes within each taxon of interest, and then it pulls these and explores the optimal set to maximize coverage across all taxa. This approach was the work of Hayden Metzke, who at the time was a graduate student in Pardis's lab, who really led all of the computational development and conceptualization of this. And our probes, as well as the tool itself, Catch, are freely available. Using Catch, we designed a probe set to cover all human-associated viral taxa using 350,000 probes. As you can see in the bar plot on the left, more diverse viruses like HIV and hepatitis C have more probes. But, as you can see from the figure on the right, the total number of probes is still markedly smaller than what would be needed using more naive approaches, such as clustering-based approaches. This example here is given for hepatitis C. And perhaps the biggest difference with Catch is that compared to earlier designs, these probes are guaranteed to capture full genomes of the viruses in the input set, assuming certain properties of probe hybridization. And this guarantee was really a kind of unique development. After designing these probes, we then tested them. And the first thing that we see is that our sensitivity to detect a virus increases by around an order of magnitude, irrespective of the host background. Crucially, not only are we better able to detect, but we do a much better job at assembling genomes from these samples. Here I'm showing clinical samples containing 8 different viruses. And I want to draw your attention in particular to Zika virus. So in orange, we have the genome lengths assembled without capture, and in blue, we have the genome lengths assembled with capture. And you see that in the 6 samples of Zika virus which we attempted, we were able to recover over 75% of the genome in 5 out of those 6 samples when we applied capture. Another key piece here is that we can do this without distorting the viral populations in these samples. And we assess this in 2 main ways. On the left, we looked at the percentage of reads assigned to each viral species, in a series of simulated co-infections that had between 2 and 8 different viruses mixed together. On the right, we looked actually within a single virus at within host variants. And this was for dengue virus. We had a number of dengue virus samples that contained within host diversity. And in both cases, we see that the proportions reflected by these variants, or either by species or by variants, maintains fairly stable with and without capture. An accurate representation of these frequencies is really critical if we want to use these types of approaches for more detailed studies. Again, not just for detection, but for starting to get into more the genomic epidemiology, looking at transmission or evolution over time, in addition to simple detection. Finally, we applied this technique to Lasse samples from the 2018 outbreak in Nigeria. And again, this was for 23 samples, and we saw a marked improvement in genome recovery for all of these after enrichment, although we didn't obtain complete genomes for all of them. These samples came from the same hospital, Irua Specialist Teaching Hospital, that I mentioned earlier in my talk. And shortly before the yellow fever outbreak in late 2018, there had been an unusually large seasonal outbreak of Lasse virus in the same area. In this case, there were so many samples that we had to ship a lot of them to the Broad for sequencing in order simply to manage the volume. But really our goal is that this work should always be able to happen locally. And I believe that capture-based approaches will be really important here, as they can substantially reduce the cost of assembling viral genomes when using something like a MiSeq, while if you use a pan-viral type panel, such as the one that I've just described, you can keep that sort of unbiased nature of metagenomic sequencing. I just want to give one example of this. This is a Lasse virus genome sequenced to approximately the same depth, with and without capture. And if we use the costs of MiSeqs, you can see that the cost is dramatically reduced when we use capture. And this is the cost really from sample receipt all the way through to genome generation and analysis. And it's not only in Nigeria that we've been applying these approaches, but through our involvement in the African Centers of Excellence for Genomics of Infectious Disease, we've also been training partners in Sierra Leone and Senegal. And all of these sites are equipped with MiceEX and have been performing genomic surveillance for detecting emerging threats across these regions. These approaches have been used for detecting a number of different viruses, including studies on Lasse virus, dengue virus, and of course most recently SARS-CoV-2. And then in spring 2020, our focus shifted too, as did many others, to the emergence of SARS-CoV-2, in particular in our own area, to understanding how the virus was introduced and spread within Massachusetts. As we'd already had considerable experience applying metagenomic sequencing to viral outbreaks across the world, we were quickly able to pivot and start sequencing almost immediately. Indeed, we sequenced almost 100% of the cases identified in the first 2 weeks of March 2020 from the Boston area. In total, sequencing over the first wave, we assembled a dataset of over 800 high-quality genomes from across the state. These were focused around the Boston area. From these, we identified over 100 independent introductions into the state over the course of the first wave from many different places. And we studied in depth 2 super-spreading events, one from a skilled nursing facility and another from a conference, that had substantially different epidemiological but also genetic characteristics. However, what I wanted to talk to you about in particular was that in addition to all this, of course, our focus was mostly on SARS-CoV-2, but as we were one of the few groups using metagenomic sequencing for this, we were also able to look at the frequency of co-infections. And what we found was that these were actually genuinely infrequent in our dataset. So from around 1,000 cases in the first wave, we found only around 1.4% of them showed evidence of respiratory co-infections. What was really great about this was that we were actually able to confirm a number of these. So while this was done in a research setting, through our collaborations with the Massachusetts General Hospital and Massachusetts Department of Health, we confirmed most of these with the BioFire assay. Of course, the caveat of this approach again is that we were not able to recover viral genomes from samples with very low viral loads. And so we began looking again towards our targeted enrichment approaches. We already knew from our earlier work in designing the pan-viral panel that these probe sets were able to capture sequences that were not part of the initial design. In that case, we'd done this using H4N4. So H4N4 is an avian influenza that wasn't part of our initial design, however many other influenzas were. And we saw that when looking specifically at H4N4, down to an identity between probe and sequence of around 80%, we still saw a substantial increase in depth of coverage after capture. We wanted to do this a bit more precisely for SARS-CoV-2 specifically, and so Hayden took the first few SARS-CoV-2 sequences, first few SARS-CoV-2 genomes that were released, and estimated that because of the similarity to previous coronaviruses that were part of the input set, around 72% of the genome would have been covered by at least one probe in our existing probe set. However, we felt that this might not be enough, and so Hayden quickly designed a new probe set with the first few SARS-CoV-2 genomes as input. We actually felt this was a good opportunity to do more than just SARS-CoV-2, and so we built a probe set that was actually 100,000 probes that covered around 20 of the most common respiratory viruses, and I'm showing those on the left here. We synthesized this probe set quickly and did some testing, and we found that indeed our ability to assemble partial or complete viral genomes increased substantially. On the right here, I'm showing a plot similar to one I showed before, where no captures in orange and captures in blue. And we see the CT values of these samples are actually written above the bars. And so with just 500,000 reads, we were now able to assemble genomes from a CT of around 31. Not only was this a really useful tool, this was actually, we felt, a really great use case of what we'd imagined for Catch, the possibility to redesign a probe set quickly with new specifications as new genomes become available. We've used this respiratory virus panel in a few different ways now. I just wanted to quickly mention one of them before I wrap up, which is in discriminating between reinfection and persistent infection. This is a focus of a lot of interest right now, and an area where genomics can be really critical. The challenge here is that standard diagnostics do not provide a way to differentiate these two events. So things like antigen and RT-qPCR tests can't differentiate the actual viral sequence in a sample. But while clinical data can sort of inform whether or not a case is likely to be reinfection or persistent infection, really comparing the viral sequence is the only true way to definitive way to discriminate between these two scenarios. So I wanted to just mention one such case that we worked on, which was an immunocompromised patient who had 2 periods of disease separated by around 100 days, and I'm showing the timeline at the top here. And as is often the case with these situations, the viral load, particularly in the second disease period, was low. However, we didn't want to use amplicon sequencing because we didn't want to bias those intra-host variant frequencies that could be useful for future studies of virus evolution over time. So we used hybrid capture and our respiratory virus panel in order to enrich particularly sequences from that second time point, that second infection period. And we found when we put these on a phylogenetic tree, which I'm showing here, the ones from the second disease period were quite distinct from the first disease period. And this led us to conclude that this was likely a case of reinfection. And this work was recently published, led by Chris Tompkins-Tinch, another graduate student in Pardis's group. So I'm going to wrap up here by just saying that I think we're in the middle of a really exciting time for the application of genomic methods for viral detection. Obviously, this didn't come from nowhere. The potential of these technologies and the importance not only of detecting, but crucially in sharing viral genomes was recognized as early as the West African Ebola outbreak. And we've certainly seen an acceleration in the application and in the availability of these approaches over the last year. But I think there's still far to go. And I think there's a key role for metagenomics to play here. We often talk about this as a hub and spoke model, an integrated surveillance system that would use rapid diagnostic tests in local clinics, molecular assay panels in regional centers, as well as genomic sequencing and particularly metagenomic sequencing in national genome centers to detect emerging viruses and rapidly iterate on designing diagnostics and identifying emerging threats. And this is something that we and others are actively working towards, both with our partnerships in West Africa and elsewhere. And when I say we, it really is a huge team of people who made all this work I talked about possible. In particular, Hayden and Chris on the target enrichment. Hayden did all of the work designing Catch and is a truly phenomenal computational scientist. Summer and Nigeria and our Nigerian partners on all of the work I presented from ISTH and Redeemers. And Jacob, Chris, Bronwyn, Danny, Pardis, and our Massachusetts partners for the SARS-CoV-2 work. And thank you very much again for the invitation to speak, and I'm looking forward to taking any questions.